All right, friends, welcome back. Um, so hopefully all of you got my email question 7 on the exam. Don't worry about it. I accidentally deleted the right answer from question 7. So all answers are now correct for question 7. You should fill in something because if you don't fill anything, I don't think the testing center will give you points. And then you and I will have to have an uncomfortable conversation about why you didn't fill in anything even though I told you to fill in just anything. Okay? Got it? So just fill in something. One answer. Any answer for question 7. Okay. So now in this next chapter, we're going to take everything we've learned so far and put it into understanding catabolism, which is how you break down molecules to obtain other high energy molecules that you can use to power life. So we have sort of 3 main sources of nutrition in our diets, and we've talked about each of these molecules in recent days. You have lipids, you have carbohydrates, and then you have proteins. All right, and of course lipids are the triacyl glycerols. Of course, there's more than just this in your diet, but this is our main sources of energy. You have carbohydrates, so we can draw one of the anomers of glucose, and of course, most of the carbohydrates we eat are polysaccharides, so starch. And then you have proteins, which are, of course, polymers of these amino acids with various side chain groups and amide bonds linking them. Okay, and the pathway for getting energy from each of these groups starts in different places, but ultimately ends up in the same place. So for example, we're going to see that carbohydrates go through a process called glycolysis, and following that there's well, we'll just call it glycolysis, and that generates ultimately 2 molecules of a group called acetyl-CoA, which is a thioester. Sorry, I guess we could say glycolysis ends in this molecule pyruvate, which then nature does something called an oxidative decarboxylation to generate a thioester. And these 2 carbons that were in pyruvate end up in acetyl-CoA. And we care about acetyl-CoA because that gets shunted into what's called the citric acid cycle, or the Krebs cycle, and this occurs in mitochondria, and the output of the citric acid cycle is CO2 and an equivalent of ATP, and also NADH and FADH2, which go to the electron transport chain, and that drives the proton gradient, facilitates oxidative phosphorylation, and then you get ATP, which you can use to drive all kinds of biological processes. Okay, and oxygen is consumed in that process, and we make water from oxygen. Okay, so that's carbohydrates in a nutshell. That's where they end up. Proteins are hydrolyzed by proteases into the component amino acids. So you've probably heard of trypsin, which is a protease that exists in the digestive system. There are others, but these hydrolyze the amide bonds in the proteins and break them down into their component amino acids. This is why, incidentally, you generally do not take protein drugs orally. For example, insulin, perhaps the longest used peptide protein medication you inject. There is not yet an orally available insulin because you have enzymes in your guts that chew up insulin into its component amino acids. And so if you were to ingest insulin, you would just be ingesting a really, really, really expensive protein shake. And that's an active area of research, how to make protein drugs less susceptible to proteases. We're working on some of that in my lab, but that's a basic problem. There are other metabolic pathways that convert these amino acids into acetyl-CoA. And we will not talk about those pathways, though a lot of them are really, I think most of them are really, really well understood. And if you want to know more about them, you can simply look it up. If you want to be overwhelmed at metabolic pathways, just Google metabolic pathway chart, and it will show you something that if you were to read it, you would probably, if you were to be able to see it and see the print, it would probably be as big as that screen there, with many tiny words all over the place and lots of molecules. Anyway, so it's pretty complicated, but we're going to keep it simple. But ultimately, if your body can either use these amino acids to make proteins in the ribosomes in cells, or it can convert the amino acids into acetyl-CoA, and then you can put those into the citric acid cycle and get ATP. Finally, the lipids. We have talked about what happens to them before. You have an enzyme called lipase, or lipases, various lipases that hydrolyze the triglyceride into glycerol, and then also fatty acids. And there are 3 fatty acids per lipid or per triglyceride, and they don't have to be all the same. Glycerol gets modified to something that can go into glycolysis, which we'll learn about. And then there's a separate pathway called fatty acid beta oxidation that can break the fatty acid down into acetyl-CoA. So this is our overview of how what you eat ends up being used by your body. And we will return to acetyl-CoA a little bit later. We'll talk about what the coenzyme A is, but for right now it's okay for you to think of acetyl-CoA as just a thioester. So a couple of things about thioesters. The negatively charged thiol is a better leaving group than a negatively charged oxygen. Therefore, here's my 3 little dots. That means therefore, I think, in math. If you're J.R.R. Tolkien, I think you use the 3 little dots to dot your I's when you're Bilbo writing his there and back again. But I'm sorry, the Star Wars references come easy for me. The Lord of the Rings, though I love that saga, the references are a little bit weaker there, so my apologies. Therefore, thioesters are more reactive in nucleophilic acyl substitution reactions. Which means that we're going to see examples where we take acetyl-CoA and some nucleophile, and we replace the CoA, the CoA thiol with the nucleophile. Okay. A second consequence is that the alpha protons of acetyl-CoA are more acidic than ester alpha protons. So that means that we can convert acetyl-CoA into an enolate, which then we can use in various aldol/Claisen type reactions as a nucleophile. Recall that the pKa of a proton alpha to an ester is about 25. For a thioester, it's lower than that. But what this means is that acetyl-CoA can do all of the chemistry we've talked about before for esters in this class, but faster and more favorably. And so it's a really versatile molecule, and it's not only used in biology for going into the citric acid cycle. You can do a lot with acetyl-CoA. I will point out, too, that your body has ways of taking acetyl-CoA and converting it into a fatty acid, so you can go backwards. You have ways of taking acetyl-CoA and turning it into a carbohydrate. That's called gluconeogenesis. And then for many of the amino acids, you have ways of making those from acetyl-CoA as well. There are a few that are essential in our diets that we can't make ourselves, but there are a lot that we can. We're going to talk about catabolism, which is breaking things down. The reverse is anabolism, which is going back to more complicated molecules. And those pathways are known. A lot of them use similar enzymes, but we're not going to talk about them in this class. All right, with that overview, we're going to start with lipid metabolism, and then we'll move through carbohydrate glycolysis and the citric acid cycle. We likely will not talk at all about amino acid catabolism. All right, so ester hydrolysis is the first thing that has to happen. You don't need to worry about copying this down. We've seen this before, and in any case, we're not about memorizing stuff that I don't tell you to memorize. So the function of the lipase enzyme is to hydrolyze each of these ester bonds to release the alcohol groups on glycerol and then 3 of the fatty acids. And the lipase enzyme active site has 3 key amino acids. If you were to look up the structure of the lipase enzyme, here are the amino acid numbers in the sequence. Notice that they're not consecutive, which is really kind of cool. When nature makes an active site, you know, the protein chain, okay, here to residue 152, and there's serine, then we're going to swing around back here. Finally, we're to 176, there's aspartate, and then winding around, and finally at 263, we've got histidine, and so on. So it's kind of complicated how nature arranges for these amino acids to be all in the right spot. What we're doing with this catalytic triad is we're going to use the serine hydroxyl as a nucleophile to hydrolyze, to attack the carbonyl carbon of the triglyceride. Ultimately, we want to replace this OR group from glycerol with water, but we're going to have an intermediate where we replace the OR group from glycerol with the oxygen from serine. Of course, you would expect, based on what we've learned earlier in the class, that a neutral oxygen, OH group, is not nucleophilic enough to attack a carbonyl carbon directly. And so the catalytic triad's purpose is to deprotonate serine so that it can attack the carbonyl carbon directly. Another thing that I think we talked about is there are amino acids that are hydrogen bonding to this carbonyl oxygen to activate it to attack. So the thing that does the deprotonating of serine is first aspartate deprotonates histidine, then histidine in turn deprotonates serine. There is an issue here. Some of you may be concerned about pKa's. The pKa of the serine side chain is, come on, 16, good. What about histidine? When it's protonated, what is the pKa of the conjugate acid of histidine? 6. And what is the pKa of the conjugate acid of aspartate. 4. So it should frankly be kind of weird that histidine is able to deprotonate serine, because the pKa's tell you that's not favorable by a factor of 10 billion. And then it also should not be that favorable for aspartate to deprotonate histidine. So there's a whole bunch of research into how this happens. 2 short answers are: first, enzyme active sites are not like bulk water, and so pKa's can change in enzyme active sites. They often are more nonpolar than water is, and so that can change the pKa's. Another explanation is something that's called a low barrier hydrogen bond that involves some quantum mechanics that we don't need to worry about. But the idea is basically that these residues are so close together that it actually makes the pKa's a lot more favorable. In any case, we get the familiar tetrahedral intermediate, negatively charged oxygen, and we're going to kick off this OR group from glycerol as our leaving group, but as it leaves, it's going to pick up a proton from histidine, which is going to pick up a proton from aspartate. So over the course of this 2 steps of the reactions, we have used these 2 residues to shuttle a proton back and forth to get a nucleophile to be more nucleophilic and to get a leaving group to be a better leaving group. At this stage, we're going to repeat the steps that were talked about previously, only this time we're going to use water as our nucleophile. Histidine will deprotonate water, aspartate will deprotonate histidine, OH minus attacks the carbonyl carbon. You get a tetrahedral intermediate, the negatively charged oxygen kicks down, and as OH minus leaves, it picks up a proton from histidine, who picks up a proton from aspartate, and then you've regenerated your enzyme and you've liberated the fatty acid. One question that people often ask is, why the heck do we have this acyl enzyme intermediate? Why not just have water from the beginning do the attacking? And the answer is, I don't know. But presumably, if there was a more efficient way to do it, nature would have done it already. And so it's possible. There's got to be some advantage to having the ester with the serine side chain, and not sure what it is. So I got nothing there. If there may be an answer, you can Google it and see what the answer is. Okay, so that liberates our fatty acid and glycerol. So let's show you what happens to glycerol. Glycerol is going to be converted to something called dihydroxyacetone phosphate, which is an intermediate in glycolysis. So what happens here is glycerol, and we're going to take ATP, and we're going to transfer one of the phosphate groups of ATP onto glycerol using an enzyme called Glycerol Kinase. Now, again, memorizing stuff isn't that useful, but for stuff that you're going to hear again and again and again, being aware of the lingo can be helpful. Kinases are enzymes that transfer phosphate groups. All right? And the molecule that we make here is called glycerol 3-phosphate. And we should briefly mention, this is a great time to just talk about this reaction of transferring a phosphate group. Here's the structure of ATP, which you have seen recently. There are 3 sort of important parts to the ATP structure. There's the phosphoanhydride portion, which in ATP you've got 3 of them. Then there's the ribose portion with the 2 OH groups down, and then you have the adenine portion. And yeah. Okay. So, and we've talked before about how we've sort of numbered these phosphate groups alpha, beta, and gamma. I will point out that often magnesium is a counter ion for these negatively charged phosphate groups. And in many cases, magnesium makes the chemistry a little bit easier. So if you look at ATP, as we pointed out before, each of these bonds between phosphate units is a phosphoanhydride bond. And phosphoanhydride bonds are weaker than phosphoester bonds in the same way that regular anhydrides, carbon anhydrides, are more reactive and are weaker than regular esters, right? And the reason for that is that if we break a phosphoanhydride bond, the leaving group is pretty stable. So what happens here is that the enzyme will remove a proton from the hydroxyl of glycerol. And especially with these enzyme catalyzed steps, we're going to do something we did before with imine formation. You'll recall that sometimes proton transfer has to happen first before a reaction can happen. That's called specific, no, dang it, I forget. Sometimes the proton transfer has to happen first. That's called specific acid or base catalysis. Sometimes the proton transfer happens during the rate determining step, and that's called general acid-base catalysis. Whether it's one or the other is sometimes kind of murky. Sometimes it's known, sometimes it's not. So we will often condense proton transfer steps in the same steps as other things, just to make our lives a little simpler. Of course, if you wanted to draw the proton transfer step, draw the negatively charged oxygen intermediate, and then move on from there, that's fine too. But to facilitate our discussion, you remove the proton from glycerol to get a negatively charged oxygen. Then that negatively charged oxygen attacks the gamma phosphorus, and the leaving group leaves. So you're going to think of all of this as being just a really good leaving group. And the reason it's a good leaving group is because it's the conjugate base of a strong acid. Phosphoric acid is our model acid, and the first pKa of phosphoric acid is pretty low. It's in the ones to twos, and so it's quite easy for this leaving group to leave, to say nothing of the fact that you've got magnesium here that's neutralizing some of the negative charge. I'm showing this mechanism as though it were SN2. I think I mentioned at some point in the past that there are some people who will draw first this bond forming to give you a penta coordinate, 5 bonds to phosphorus, 5 atoms bonded to phosphorus intermediate, and then the leaving group leaves. To keep things simple, I'm going to show it to you in SN2 fashion, though if you get to a biochem class and say Professor Wood shows you first the electrons kicking up here so that you now have 5 groups around oxygen, and then an intermediate where the electrons kick down and kick off the leaving group, that's fine too. We're just going to keep it simple. And so that generates that SN2 reaction, gives us glycerol 3-phosphate. And notice that we have replaced a weak phosphoanhydride bond with a stronger phosphoester bond. So this is downhill in energy. And this is one of the ways that ATP fuels reactions. You can, because ATP hydrolysis in and of itself is thermodynamically favorable, so you can often couple it with other reactions to drive them forward. But in this case, the product is more stable than the starting material. People will talk about how ATP breaking these phosphoanhydride bonds is favorable, and again, the analogy with carbon anhydrides is useful. The carbon anhydride is less stable than the carbon ester in the same way that a phosphoanhydride is less stable than a phosphoester. But one other thing that makes this reaction favorable is that you have all these negative charges together, close together, and when you hydrolyze the phosphate group on the end, you get rid of two of them. And so there's some electrostatic repulsion that makes the phosphate transfer thermodynamically favorable. So we're going to see stuff like this again and again where we use ATP to make a phosphoester. The leaving group here, by the way, is ADP, which I'm not going to draw, but there it is. Okay, questions so far? So the next thing that happens to glycerol 3-phosphate is an oxidation step. And so here is glycerol 3-phosphate. And I will abbreviate the phosphate group like that. You're welcome to do that too. We're going to oxidize this with NAD+. And I'm going to write this the way biochemists do, even though we know there's no such thing as a proton floating around in water on its own. This enzyme is called glycerol phosphate dehydrogenase. And dehydrogenases are enzymes that catalyze redox reactions, oxidation/reduction reactions. And what this is going to do is it will oxidize the secondary alcohol of glyceraldehyde 3-phosphate to a ketone, and this becomes dihydroxyacetone phosphate, which is an intermediate in glycolysis. Okay, so I think we have seen NADH reductions before, but perhaps not an NAD plus oxidation. Recall that reducing reagents in organic chemistry deliver hydrides to carbonyl compounds, like lithium aluminum hydride could reduce a ketone to an alcohol. But we're going to go the other way. We're going to take NADH and we're going to steal a hydride from this molecule to turn it into a ketone. So I want to show you that mechanism. I won't draw the whole structure of NAD plus or NADH. You can look that up, and I won't ask you to memorize it either. Here is the sort of business end of this molecule. And NAD stands for nicotinamide adenine dinucleotide. So it turns out there's a ribose here, a couple phosphate groups, another ribose, and then the adenine base. But this is the portion of the molecule that does redox chemistry. And you can sort of see how it might be able to do that if you consider the two resonance structures, consider another resonance structure available to this molecule. You could put a positive charge on this carbon, the one that's sort of para to the nitrogen, and leave the nitrogen neutral. The other thing you notice about NAD plus is you've got aromaticity. And that's the fact that NAD plus is aromatic helps control the oxidation potential and helps make it favorable for NAD plus to participate in oxidation reactions and for NADH to participate in reduction reactions. So the mechanism of this oxidation step is going to start with glyceraldehyde 3-phosphate, and you're going to have a base probably from the enzyme, remove a proton from the secondary alcohol. We're not going to draw an intermediate there. We could draw a negatively charged oxygen. But we're going to have electrons from that oxygen-hydrogen bond kick down to form a new pi bond, and then we're going to have hydrogen leave with its electrons. Now, that should make you feel, I don't know, icky, because H- is a terrible leaving group. But understand that H- never really exists here. The enzyme holds this molecule so close to NAD plus that it's sort of a direct hydride handoff. Hydride never feels like it's on its own. It's handed directly from one carbon to another. And where does it go? Well, it goes right where this positive charge is. And I mean, there's already a hydrogen there. It was implied, not shown. It's going to go right where the positive charge is. If you want to draw the mechanism over here, you can do that. It just requires a few more arrows. But doing this gives you the ketone, and it oxidizes, or it reduces NAD plus to NADH, which I'll just draw. It's going to look just like NAD plus, only there's going to be 2 hydrogens on this paracarbon. So what's with the hydride that got delivered? That's here in the NADH. Where is the proton, this H+? Well, that's the proton that was removed by the base. And that's why I hate it when they do comma proton, because it's not like that proton just hopped off and went out into the universe. A base removed it. But that's sort of how biochemists and biologists keep track of things, so that's okay. Questions about that reaction? Yeah?
So the green, it was aromatic before, and then after that happens, it's just no longer aromatic.
Right.
Is that unfavorable? I mean, I guess obviously it's not unfavorable because it happens, but why is that not unfavorable?
So why is it not unfavorable? It's a great question, and a simple sort of hand-wavy way of saying it is, well, the molecule's no longer positively charged. So whereas nitrogen had a positive formal charge here, now it's neutral. So that's favorable, but you're giving away aromaticity. So it's kind of balanced. In terms of how favorable the reduction is on its own, a reduction is only a half of a reaction, right? If you look at this, NAD plus gets reduced to NADH, but glycerol 3-phosphate gets oxidized. So you have both things together, and there is a way to compare the favorability of various reduction reactions. It's called a reduction potential, and you usually do it relative to a standard reaction, which I'm not sure what it is. So basically, I can't really tell you whether it's favorable versus unfavorable, in the sense that depending on the reaction, we can go either way. And likely the equilibrium constant is pretty close to 1 in many of these cases. Likely the favorability of oxidizing one thing is pretty well matched with the unfavorability of reducing another, or vice versa. That's really hand-wavy, but yeah. Okay, what else? All right, so those are two, in these first 2 steps, those are 2 classic examples of reactions that we will see again and again, phosphate transfer and reduction of NADH and oxidation of something else. Okay, so that's the fate of glycerol. Now let's talk about the fate of the fatty acid. All right. So, and this is called fatty acid beta oxidation. And it involves a precursor step, and then 4 additional steps that are repeated again and again. So first, we've got to convert the long-chain carboxylic acid into a thioester. Okay? So we're going to use ATP to do this. Gonna get sick of drawing ATP before the end. Can we just do that for adenine? Is that okay? Yeah. All right. Again, you've got alpha, beta, and gamma phosphates. In this reaction, you'll have magnesium here as the counter ion. Unlike the reaction we saw above, this carboxylic acid is going to attack the alpha phosphate, and the leaving group is going to be this pyrophosphate group. And sometimes when you need a reaction to have a little bit of an extra kick of thermodynamic stability, favorability, this is an interesting way to do it. And the reason for that is that once you, and this is sort of an SN2 style reaction, you're going to create this molecule. And so you've converted a carboxylic acid into something that looks kind of like an anhydride. So the bond that we're breaking is a phosphoanhydride bond. And now we've got what's called an acyl phosphate, or sometimes I call it a mixed anhydride, because it looks like an anhydride, only the thing on this side is different from the thing on this side. But the effect really has been to put a good leaving group on the oxygen of the carboxylic acid. Now, what about pyrophosphate, that other thing that was the leaving group in this SN2 reaction? This is our this is sort of the secret weapon in this reaction. Biochemistry texts and biology texts will abbreviate this as PPI. This is a secret weapon because it spontaneously undergoes a hydrolysis reaction that converts the pyrophosphate into 2 phosphate groups. Oops. And I've drawn them in the form that they would sort of predominantly exist at pH 7, probably a mixture of this and the conjugate acid. These are both would be called PI for inorganic phosphate, but this is downhill in energy, quite favorable. And so, and the first step here of breaking this bond is also favorable. So this sort of drives the reaction even further because you've got a further thermodynamic sink to go down, and that'll push the reaction forward. So this is your acyl phosphate, and then in the next step, we're going to replace that good leaving group with coenzyme A. All right, so I'm going to copy this acyl phosphate. And I'm going to highlight in green the good leaving group, which is going to leave as AMP. And then we have coenzyme A, which has a sulfur and a hydrogen. I'm not drawing the structure of coenzyme A for you. We can do that another time. This is going to be nucleophilic acyl substitution. So the mechanism should be unsurprising. A base removes a proton from the thiol. The negatively charged sulfur then attacks the carbonyl carbon to give you a tetrahedral intermediate, which I'm going to abbreviate like this. And then I'm just going to put AMP here. And then electrons kick down and kick off the AMP leaving group to give you now your fatty acid thioester plus AMP. Okay, see what's going on there? Just regular carbonyl chemistry, nucleophilic acyl substitution. And if you take a step back and look at what we've just done, we've taken a carboxylic acid that was negatively charged and didn't have a leaving group on it, and we replaced that with, we used ATP to turn that oxygen into a good leaving group, and then we're swapping out the AMP for something else. Now, you may be wondering, why not just stick with AMP if that's a good leaving group? And I don't have an answer other than that thioesters are the reactions of thioesters are really fast. It's really fast to swap out one thiol for another, and it's also fast to swap out some other nucleophile for this thioester. And we're going to see that and then the other reason for having CoA here is because we ultimately want to get acetyl-CoA that we can take into the citric acid cycle. All right, so that's preparation. Now we'll get to the steps that are repeated. So, yeah. But, yeah, sorry, questions so far? All right. So here's your fatty acid thioester. And our goal is going to be to clip off a 2-carbon unit as acetyl-CoA. And so to actually show this, we probably need to draw a few more carbons of the thioester. 1, 2, 3, 4. That's good enough. All right. So and here's the alpha carbon, and here's the beta carbon. If we want to clip off a 2-carbon unit, we want to break the bond between alpha and beta carbons. If you want to break the bond between an alpha and beta carbon, there's a couple reactions you could be thinking of. One is retroaldol, and the other is a retroclason. But both of those would require some kind of oxygen on the beta carbon. All right? But we don't have that. So our first task is going to be to oxidize the beta carbon so that we can then get to an aldol or clason product and go backwards. But we don't have that oxygen there. So the way we're going to do this is we will have a base remove the proton alpha to the carbonyl. This is just enolate formation. And remember, I told you that the proton alpha to the carbonyl is more acidic than a proton alpha to an ester. So forming this enolate is quite easy. Here's the negatively charged alpha carbon. and then here are 2 hydrogens. Now, we're going to need to do an oxidation reaction. And what that would mean is ideally we want the electrons on this enolate to kick down, and then, sorry, something on my screen is asking if I'm still here, which I wonder about, but I think I am. We want to kick off hydride as a leaving group. Of course, hydride is not a great leaving group, so we need something to receive it. And the thing that's going to receive it is another of nature's oxidizing agents, FAD. FAD is a little bit more complicated than NAD+, though ultimately it's going to do the same thing. I'm going to draw just the important part of the molecule. So this is a squiggly line leading to a ribose sugar. We're not going to worry about that at any level of detail. I will not have you memorize the structure of FAD or FAD+. So don't worry about that. And I will just show you how the arrows go, and we won't actually worry about why this can happen, other than that you have electron withdrawing groups here in these oxygens that are conjugated to this nitrogen and that make that nitrogen electron poor. So the hydride can be handed off directly to that nitrogen, and at the same time we can push a few other arrows to get to a neutral product. This is some acid from the enzyme, but if you look at what we've done then, that will oxidize what used to be a hydrocarbon to something that now has a double bond. And then we will also make FADH2, which looks like this, and this is the reduced form. FAD and FADH2 are used for a lot of different things, not just this oxidation reaction, but we're not going to worry about that. This is the reduced form. F stands for flavin, though, and you get this cofactor from riboflavin in your diet. Okay, so this is an oxidation. The alpha-beta unsaturated carbonyl compound got oxidized, and FAD got reduced to FADH2. Okay. So remember how I said our goal is to get an oxygen on the beta carbon? You tell me, if you were going to do this in the lab, what you would do. What kind of reaction could you do to get an oxygen on the beta carbon? Okay. I'm hearing some whisperings, but a reluctance to commit. Alpha-beta unsaturated carbonyl compounds are electrophilic where? Of course at the carbonyl carbon, but also at the beta carbon, right? Because you can draw a resonance structure where you put negative charge here, double bond here, and positive charge on that carbon. That means a nucleophile can attack the beta carbon. And what nature uses to do this is simply water. So this is a conjugate addition reaction. It's just like the Michael reaction, only instead of a nucleophile, we're using, forget what step we're on for. Instead of an enolate as a nucleophile, we're using the OH group of water. Okay, so that's as far as we're going to get today. There are yet 2 more steps, and then we'll be done with fatty acid beta oxidation. So, good luck on the test, and I will see you Friday.